Welcome to the virtual tour of our poster. In this work, we study the contrast sensitive function in machine vision. The CSF is a fundamental signature of any visual system by measuring the visibility threshold at every spatial frequency. It has been extensively studied across several species in the animal kingdom. The CSF that emerges in all the studied animals looks like an inverted U-shaped function. Given in the past couple of years, the artificial visual system has reached a level of performance in several visual tasks. The interesting question is whether the underlying representation learned by those networks also resembles the biological vision. Given the importance of CSF, in this study we wondered how the CSF of deep neural networks look like. To answer that question, we systematically studied the ResNet architecture that consists of 5 areas and can be trained for any type of visual task such as scene segmentation, object classification, image compression, etc. Once a network has been pre-trained on one of these visual tasks, in order to measure its contrast sensitivity, we add a linear classifier on top of each area. This linear classifier, we refer to it as a contrast discriminator that is only exposed to natural images similar to those. The images are multiplied by a Gaussian to remove any sharp borders. And during the training of the contrast discriminator linear classifier, the pre-trained weights are frozen. Once the contrast discriminator linear classifier has been trained in order to measure its CSF, we follow a procedure similar to human psychophysics by inputting the networks with 2 images of one uniformly gray background and another sinusoidal grating. At every spatial frequency, network is input with 56 trials. The contrast of the sinusoidal grating is adjusted till the network reaches 75% accuracy, which is also a threshold for the human contrast sensitivity function. In order to compare the CS of the networks to the humans, we made an assumption that the networks matches the entire fovea, which is of 2 degree and 60 cycles per degree. To cover this, we need an images of 256, which gives us a maximum 228 cycles per image. Let's look at some of the results. In this figure, we see the contrast sensitivity function of the 4 networks across 5 different areas. At every insert, the x-axis is the spatial frequency, the y-axis is the sensitivity. The green curve is the segmentation network trained on the MS-COCO datasets. The orange object recognition on ImageNet. The blue is a variational autoencoder, and the red curve is a contrast discriminator network trained from the scratch without any pre-trained task. The comparison of the CSF across areas and networks reveals that in area 2, 3, and 4, the segmentation, classification, and compression networks obtain a CSF that very well resembles the human CSF. Contrary to that, the contrast discriminator network trained from the scratch without any pre-trained visual task doesn't obtain the human-like CSF. This can be considered as a control experiment showing the CSF of those networks is not a byproduct of the contrast discriminator linear classifier and is truly the features learned by those networks in the pre-trained tasks such as segmentation and classification. In order to measure the degree of the similarity, we computed the correlation between those curves and the human CSF, which is reported in this table. It seems the segmentation networks obtain higher correlation in comparison to the other. Why the nature of a visual task might decide the shape of the CSF requires further investigation, but it might be related to the fine granularity of the task. We further investigated the environment of the pre-trained task on the obtained CSF by training 3 object classification network on ImageNet that are either high-pass, unfiltered, or low-pass images. The comparison of the CSF across these 3 different regimes of the networks reveals the CSF of the high-pass network is clearly shifted towards a high spatial frequency. We further trained the object classification network on aerial images similar to what a falcon and eagle might see, and again we see the peak of the contrast sensitivity function is shifted clearly towards the high spatial frequency which matches very well the contrast density function of those pairs. The comparison of the unfiltered and the low-pass regimes shows that both networks can obtain human-like CSF, but there are two fundamental differences. The unfiltered images, the CSF is noisy and it does not emerge from the first epoch, and it requires dozens of the epochs to reach that. Contrary to that, the human-like CSF appears from the first epoch in the low-pass filter. It remains solid constant across several epochs. We further conducted different control experiments across different instances with different kernel size, and the CSF of the low-pass regime remains unchanged. This suggests the low-pass images, similar to the optical modulation that occur in the retina, might have a great impact on our CSF. The source code of all our experiments are available on our GitHub. Thank you for your listening. Looking forward to all the comments and suggestions.